The modern genetic code is one of the most refined and complete information systems found in nature. Built from 4 nucleotide bases, adenine, cytosine, guanine and thymine or uracil in RNA, arranged in triplets, it yields 64 possible codons. These codons specify the 20 standard amino acids and the stop signals necessary for protein synthesis. This structure is not an arbitrary relic of evolution. It is the product of billions of years of selection for stability, resilience, and adaptability, and it is rooted in the combinatorial completeness and geometric coherence of triadic encoding. Each codon is a 3-position base 4 sequence. 4 possibilities at each position produce 64 combinations. This creates a closed, symmetric space in which every possible triplet exists. That completeness is not just numerically satisfying, it is functionally essential. It allows for degeneracy in the code, where multiple codons can specify the same amino acid. That redundancy provides error tolerance. A single nucleotide mutation often leaves the protein unchanged or only subtly altered, preserving function and buffering the organism against genetic noise. It also enables compatibility with overlapping reading frames, a feature more common than many assume, in which the same stretch of nucleotides can encode multiple proteins when read in different frames or from opposite strands. The result is a code that is robust, adaptable, and capable of sustaining self-replicating complexity across evolutionary timescales. Yet in recent years, synthetic biology has set out to rewrite this foundation. High-profile efforts such as the SYN61 and SYN57 strains of E. coli have attempted to reduce the number of codons in the genome. The stated goals are both practical and experimental: to free unused codons so they can be reassigned to synthetic amino acids, creating new chemical capabilities, and to test the limits of how far the genetic code can be compressed while still producing viable life. These projects have pushed genome engineering into unprecedented territory, requiring tens of thousands, and in the case of SYN57, over 100,000, precise genome edits. The recoding process is slow and laborious, and each section of the genome must be tested for viability before proceeding, because a single faulty change can derail an essential function. The technical difficulty is immense, but the deeper problem is structural. The genetic code is not a flat table of interchangeable triplets. It is a tightly interwoven architecture where each codon participates in multiple functional layers at once: protein coding, regulatory control, RNA folding, translational efficiency, and in many cases multi-frame or overlapping usage. One of the most acute obstacles faced in SYN57 was the problem of overlapping frames. DNA codons consist of 3 genes read sequentially, but groupings are determined by the start and direction of the process reading the strand. So nearly every gene is included in 6 possible reading frames. Nature exploits this economy, especially in compact or highly regulated genomes, by encoding multiple proteins using the same nucleotide sequences. In such cases, changing a codon in one reading frame may inadvertently introduce a stop codon, alter an amino acid, or disrupt a regulatory signal in another frame. Reducing the codon set magnifies this problem. With fewer triplets available, the combinatorial space that makes such overlaps possible shrinks dramatically. The SYN57 team was forced to refactor overlapping genes, redesigning them so each could be recoded independently without destroying the other's function. This was not a minor technical nuisance. It was direct evidence that the redundancy and completeness of the 64 codon system are not cosmetic. They are structural, load-bearing elements that hold the genomic lattice together. Remove them, and the whole architecture becomes more fragile. Even in non-overlapping genes, codon reduction disrupts the subtle balance of translation. Codon usage in natural genomes is biased. Some codons are preferred because they match abundant tRNAs, regulate translation speed for proper protein folding, or influence mRNA secondary structure in ways that control ribosome binding. Removing these codons forces substitution with less optimal ones, which can slow protein synthesis, misfold proteins, or break finely tuned expression patterns. These changes come with an even more serious form of risk: one that is often underestimated. Genome-scale editing on this scale inevitably introduces instability, and instability in a living system is not static, it evolves. Tens of thousands of edits cannot all be fully understood in functional terms. Sequencing confirms the changes made, but it does not reveal every context-dependent effect, every long-range interaction, or every role that a codon plays beyond its protein coding meaning. RNA structure, antisense transcription, hidden regulatory elements, many remain unmapped. When stability is compromised, evolution becomes the wildcard. The organism will change in ways no one has modeled and no laboratory control system can perfectly contain. The 64 codon code's resilience is rooted in its completeness. It fills the entire combinatorial space available to a triplet base 4 system, giving life the maximum possible diversity of coding triplets while preserving a lattice of redundancy that buffers against error. This is a closed, symmetric system in which every possible state is present. In symbolic terms, completeness is what allows such systems to generate internal complexity, maintain coherence, and evolve stable substructures. Removing codons breaks that closure, introduces asymmetry, and collapses the richness of possible internal relationships. This is why the 64-ness of the code matters. It is not merely a count of possibilities, it is the natural resolution at which the system achieves stable complexity. Below it, the lattice begins to lose both strength and flexibility. Degeneracy collapses, neutral mutation networks shrink, overlapping coding becomes harder or impossible, and the space for safe evolutionary exploration contracts. The risk here is not simply technical failure, but epistemic failure. By treating the genetic code as a set of parts that can be swapped or removed at will, these projects risk overlooking the very features that give the system its stability. Codon reduction is not just freeing up space for novelty. It is removing the hidden bolts that keep the structure from coming apart. This is not simplification, it is the dismantling of a complete evolved architecture. And because the genome is not static but subject to constant evolutionary forces, instability introduced today will not stay in the form it was engineered. It will mutate, recombine, and spread unpredictably. The 64 codon system is not good enough by chance. It is the minimal complete code that could arise and persist under the chemical and informational constraints of life. Reducing it strips away built-in safeguards and narrows the pathways through which life can adapt and repair itself. This is not a theoretical worry, it is an observed reality in codon reduction experiments. Fewer codons mean less error tolerance. Fewer codons mean reduced adaptability. Fewer codons mean greater fragility in overlapping and multi-frame coding. Large-scale edits produce emergent effects that cannot be fully anticipated or controlled. In the end, recoding life's alphabet to remove codons is not controlled optimization. It is experimental demolition of an evolved, closed system. The genetic code's completeness is not a curiosity. It is the very reason it can support stable, self-replicating complexity. Disrupting it without understanding all of its interdependencies is a gamble with the load-bearing structure of life itself. And in living systems, once you loosen those bolts, you cannot predict how or how quickly the structure will shift.